Let's take a closer look at carbohydrates. Carbohydrates are made up of the elements carbon, hydrogen, and oxygen. There are three classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are single sugars. The examples are glucose, fructose, and lactose. Notice how they end with the suffix "ose". "Ose" means sugar. So those are the three monosaccharides that we will come across in the human body. Disaccharides are two sugars that are held together by a covalent bond. The examples are maltose, which is found in barley, wheat, sweet potato; sucrose, which is found in cane sugar, beets, etc. Our table sugar is sucrose. Lactose is found in milk. Polysaccharides are several monosaccharides held together by covalent bonds. The most important polysaccharides are starch and glycogen. Glycogen is a stored form of energy in the liver. It's basically many glucose units held together by covalent bonds. The starch is the dietary form of polysaccharide. It's found in rice, pasta, bread, etc. The main function of carbohydrates is to provide us with energy. Let's take a look at the composition of disaccharides. The word disaccharide can be broken down into di, meaning two, sachar, meaning sugar, and ide means unit. So maltose, which is a disaccharide, can be broken down into two glucose units. Sucrose is broken down into glucose and fructose. So when you eat table sugar, that's broken down into glucose and fructose. Lactose is broken down into glucose and galactose. So as we can see here, glucose, fructose, and galactose are the monosaccharides. Sugar is ose, and remember, di is 2 and mono is 1. Sugar is a problem when consumed in large quantities. Sucrose, which is table sugar, when broken down by our digestive system, becomes glucose and fructose. It's basically 50% glucose and 50% fructose. The glucose is usually not a problem because majority of the cells in our body can use glucose as a source of fuel. So that's not a problem. However, fructose cannot be used by all the cells in our body. It goes straight to the liver and most of it is converted to fat. So sugar becomes fat in the end via fructose. And as a result, fructose is the culprit and causes many problems such as diabetes, obesity, high blood pressure, fatty liver disease, and cancer. And remember, cancer cells love fructose. When you consume milk, you're consuming the milk sugar lactose. And this lactose is broken down into glucose and galactose by your digestive system. And these are monosaccharides, and it is in this form that the digestive tract can absorb it. If lactose is not broken down, then it causes a lot of discomfort in the digestive tract because it cannot be absorbed. People who are lactose intolerant do not produce an enzyme called lactase. Lactase is the enzyme that helps us to break down lactose into glucose and galactose. Ase means enzyme. Not everyone produces a sufficient quantity of this enzyme, and so that results in a condition called lactose intolerance. Let's move to proteins. Proteins are made up of carbon, hydrogen, oxygen, nitrogen, and sulfur. Proteins are made up of amino acids, which are their building blocks or monomers. There are 20 different types of amino acids, which in different combinations make up different kinds of proteins, which is why proteins are so varied. These amino acids are held together by covalent bonds, and these covalent bonds are called peptide bonds. Each amino acid has a very specific structure. They are similar in many ways. Each amino acid will have an amino group, an acid group, a carbon, and a hydrogen. It also has something called an R group. The R group is what differentiates the 20 amino acids because each R group is different. So there are 20 different R groups. So here's your amino group, which is NH2, and the acid group, which is COOH, and which is why we call it an amino acid. As we've just mentioned, proteins are very diverse and they carry out a wide variety of functions. There are 6 different protein classes and each class carries out a different function. Structural proteins provide structure, as in the case of keratin, which provides structure to hair and nails. Receptor proteins act as gatekeepers on a cell membrane, as in the case of channel proteins that allow only certain ions or molecules to enter the cell. The third type of protein are contractile proteins. Their function is to help a cell to contract and relax. This is found in the muscle tissue, and muscle cells have actin, which carries out that function. Transport proteins, as in the case of hemoglobin, transport gases such as oxygen and carbon dioxide in the protective proteins protect us from infections, as in the case of antibodies. Antibodies are proteins and they are produced by white blood cells and they travel around in the blood looking for the infectious agent. Enzymes are proteins that speed up reactions. Lactase, as we just saw, is an enzyme that helps to break down lactose into monosaccharides. Lactase is found in the digestive tract. Based on how a protein is folded, it exists in 4 different forms: primary, secondary, tertiary, and quaternary. A primary structure, the amino acids are held together by peptide bonds, which are basically covalent bonds. Usually when a protein is just made, it exists in the primary form. The next level is the secondary structure, and it's a little bit more complex. Keratin in hair exists in this form. The pink circles that you see are amino acids, and the dashed lines are the hydrogen bonds that hold this structure. The next level is the tertiary structure, and an example of this would be lactase. Lactase exists in this form, in the tertiary form, and its shape is extremely important to carry out its function. This tertiary structure is held together by interactions between the R groups that we mentioned earlier. So let's apply what we have just learned to a real life situation. We just learned that secondary structure of a protein is held together by hydrogen bonds. And the example that we gave for a secondary structure was keratin. And keratin is the that makes up hair. So let's situation. Here's a person, and in this person, the keratin has many more hydrogen bonds than a person. So there are many more hydrogen bonds that hold this keratin structure together. That's what makes the hair curly. Here's naturally straight hair. This person, keratin here, has very few hydrogen bonds. Now let's say I have curly hair and I want to straighten it. What would I do? I would use a straightening iron that basically applies heat to the hair. What the heat does is to break up these hydrogen bonds. So if you break these hydrogen bonds, your hair can now be temporarily until these hydrogen bonds reform again. So let's say I have straightened my hair and I feel good, I look good, and I step out and it's raining. And I've forgotten my umbrella. So what's going to happen is that rainwater enhances hydrogen bonds. Rainwater is attracted to the dry hair and enhances the hydrogen bonds. And as a result, a single cell contains thousands of enzymes, and enzymes exist in the tertiary structure. As we remember, the tertiary structure is held together by H bonds and R interactions. These are weak bonds and can be easily broken down by high temperature and changes in pH. Let's take the example of lactase. Lactase is an enzyme that speeds up the reaction of breaking down lactose into glucose and galactose. Lactase has a very specific shape, and if this shape is lost, it loses its function. This shape is maintained by, again, the hydrogen bonds and the R interactions. If the temperature, body temperature, increases beyond 40 degrees Celsius, then there is a possibility that these bonds will break. The greater the temperature beyond 40 degrees Celsius, the faster the breakdown of this shape. A low pH such as acid and high pH such as base can also disrupt the shape of the enzyme. And once the shape is lost, its function is lost as well. This is extremely important because the body maintains a specific range of temperature and pH, because if this does not happen, all proteins will denature and death is the result. So far, we have looked at primary, secondary, and tertiary structures. Now we'll look at the quaternary structure, which is basically two or more polypeptide chains connected to each other by weak bonds. So here we have 4 polypeptide chains, and these are connected by H bonds and R interactions. A very good example we know about carbohydrates. Carbohydrates made up of the elements carbon, hydrogen, and oxygen. The 3 classes of carbohydrates are monosaccharides, disaccharides, the main function of carbohydrates. Let's recap what we know about proteins. Proteins are made up of the elements carbon, hydrogen, nitrogen, and sulfur. The building proteins are amino acids. They are the monomers. Proteins carry out a variety of functions. So the 6 classes of proteins represent the functions that the proteins carry out. So we have receptors, which are found on membranes, enzymes such as lactase, protective proteins such as antibodies, contractile proteins found in the muscle, actin is an example, transport protein, which is hemoglobin, which transports oxygen and carbon dioxide, structural proteins such as keratin that is found in hair. The third group of biological macromolecules that we will look at are lipids. Lipids, oxygen, and phosphorus. Fatty acids and glycerol. There are three classes of lipids. We have phospholipids, triglycerides, and steroids. Phospholipids, as the name indicates, contain phosphate groups. A single phospholipid has a head and two tails. The head contains the phosphate group and the glycerol. The tails are made up of fatty acid chains. The main function is natural. It makes up the bulk of each cell membrane of phospholipids. The second class of lipids are triglycerides. This is what we call fats. The main function of a fat is to store energy. So when you run out of glucose for fuel, we use fats or fatty acids as our energy source. So you have your glycerol and three fatty acids, the three structures that you see there. The main function, and fats also form cushioning between organs. The third class of lipids are steroids. Many of our hormones, such as testosterone, cortisol, aldosterone, they're all made up of steroids. So hormones are steroids. Also, cholesterol is a steroid. Cholesterol is found in the cell membrane. The fourth class of biological macromolecules are nucleic acids. The nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The building blocks of nucleic acids are nucleotides. Each nucleic acid is made up of a phosphate, a sugar, and a base. There are four kinds of bases: adenine, guanine, thymine, and cytosine. The phosphate is the round structure that you see. Then we have the 5-carbon sugar, which is connected to the base. There are four types of bases found in adenine, guanine, and cytosine. So there we have it. A single nucleotide is made up of a Nucleic acids are made up of three classes: DNA, RNA, and ATP. DNA is short for deoxyribonucleic acid, and it is double-stranded. It looks like a double helix. Imagine a spiral staircase. The handrail of the staircase is made up of the sugar and phosphate groups, and the actual stairs that we step on are made up of the bases. The A matches with T and G matches with C. That is how you make each step. The lines that you see between them are the hydrogen bonds. The main function of DNA is to carry genetic information. DNA codes for proteins. The next class is RNA, or ribonucleic acid. This is a single-stranded molecule. If it were to match with itself, then A would match with U, which is uracil, and G would match with C. Again, you find hydrogen bonds between these matched pairs. The main function of RNA is protein synthesis. The third class is ATP, which is short for adenosine triphosphate. We have an adenine group, an OS, the adenosine, so OS is the sugar, and the triphosphate, which stands for 3 phosphates with high energy bonds between them. The main function of ATP is to act as the energy currency of the cell. Let's quickly recap what we know about lipids. Lipids are carbon, hydrogen, oxygen, and phosphorus. Are fatty acids and glycerol. You can have either 2 fatty acids and 1 glycerol or 3 fatty acids and 1 glycerol. There are 3 classes of lipids, and they are phospholipids, steroids, and triglycerides. Phospholipids, remember, those are the ones with a head and 2 tails. Earlier are hormones and cholesterol. The third are triglycerides, which are also known as fats, and these are made up of acids, and their main function is stored energy. Let's recap what we know about nucleic acids. Nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The building blocks of nucleic acids are nucleotides. Each nucleotide is made up of a sugar, a phosphate group, and a base. You could have 4 different bases: adenine, guanine, thymine, or cytosine. The 3 classes of nucleic acids are DNA, RNA, and ATP. DNA is to carry genetic information. RNA is to carry out protein synthesis.